here's just briefly my conflict of interest, and just going to proceed quickly through this. So, I mean, most of the people at ICT are very much familiar with the bumpy history of gene therapy, and specifically for the treatment of hematological diseases and disorders. And I just want to briefly touch on this because it's pretty much in everybody's mind. In the 90s and early 2000s, we had a lot of complications where actually patients died from adenovirus injections because of an immune response, as well as multiple infants who actually were treated for SCID. So just 20 years later, actually, people realized that even if it was discontinued temporarily, that gene therapy is actually a really great tool to actually continue developing. So just the fact that we have more than 40 million patients who actually could benefit from gene therapy attracted a lot of companies. And if you look up the news, it's pretty much every month you can see a new gene therapy company founded. So, and the co-development of new viruses that are safer, the so-called third-generation viruses, as well as the nuclease, actually helped a lot in this field to advance and solve some of these initial problems. So, and the first success stories were actually reported here in 2017. It's not a stem cell mediated treatment, but it's basically a CAR T-cell gene therapy approach. So the FDA has also given a green light again to actually start with gene therapy in patients again. And just recently, last year, I think it was in June or July, the first CRISPR-treated patient actually was reported, in this case for sickle cell anemia. So we are actually on the upward slope, and it's actually really quickly picking up. But the question that's always in the room is like, why is gene therapy not a routine treatment yet? And there's many, many problems actually. And one of the biggest one is actually the money. It's really expensive. So all the virus, cytokine, consumables, and the labor are very expensive. And the reason for this is mostly because they have to be performed in very highly specialized GMP facilities. And I'd like to show this map on the right bottom here. So those red dots indicate countries. I think that's a map from like 2015, a little bit outdated. But these countries actually have GMP facilities capable of doing ex vivo stem cell gene therapy. And this is a map for HIV patients who could actually benefit from a CCR5 knockout with gene therapy, but the patients are actually not in the location where the GMP facilities are. So there's a huge disconnect. The GMP facilities are currently required. So this is one of the big bottlenecks. Another one is actually in the strategy itself. So as for stem cell gene therapy, or like, let's say as an example, the treatment of red blood cells here in this hierarchy on the left bottom, you wouldn't want to really treat the erythrocytes because they are really short-living. They constantly get replaced, so your treatment would be useless after a couple of days. So what you actually want to do is you want to target or introduce your vector, in this case a virus, a zinc finger nuclease, or a CRISPR, into the self-renewing stem cell on top of this hierarchy, because this modification is then getting passed on into all the progenitors and ultimately into the mature cell type. So we are not really concerned about off-targets in many cases, because if you do erythrocyte-specific modification, it has macrophages or dendritic cells because these genes are shut down. So this way you can, for example, envision a gene transfer, for example, introducing a healthy IL-2 gene in SCID patients. You can do gene editing, as I mentioned, a CCR5 knockout for HIV treatment, or you can also actually produce CAR T cells, something that not many people focus on. But basically, the T cells are downstream of the stemma progenitor cells, so you can create stem cell-derived CAR T cells. So one of the issues that I mentioned is the strategy itself is right now, the current gold standard is using CD34 positive cells. It's cutting out a lot of these progenitors and more mature cell types on the bottom. But here, color-coded in red, green, and blue, it's actually still a mix of very different types of progenitors. And I'm just trying to highlight this here on the left bottom. The cells that we are actually interested in targeting in green, they are really the minority. So what I've been doing when I went to the lab at Hans-Peter Kiem's lab at Hematology, I was looking into strategies to increase the targeting efficiency of these cells, reduce the cost of modifying agents, and also increase the predictability. And all this work has been done primarily in the monkey. And we added 2 additional cell surface markers that are well known actually since the 90s, CD90 and CD45RA, to subdivide the CD34 positive cells in 3 fractions. And then using lentiviral vectors to modify them and gene mark them, the one population got a GFP, and one an M. cherry, and another one an M. cerulean. And we actually co-transplanted these cells into the monkey, where we can then longitudinally track the development and identify the population of interest that we need to target for gene therapy. So this is kind of like a history of like what this literature is saying on the right top. You can see that the green cells, the green box would contain the CD90 positive cells and the more primitive stem cell enriched fraction. The red triangular section would have the multipotent progenitors with the erythromyeloid lineages. And then on the left bottom, you would have the lymphomyeloid lineages with the neutrophils that are gaining 45RA. So when I did these experiments, the expectations were pretty clear because we know from transplantation that neutrophils are the first cell type coming back. They are derived from the very quickly from the LMP in the blue area. So I was expecting a wave of blue cells initially, with then a short period later replacement by red cells because the progenitors are short-living and need to get replaced by the next higher progenitor. So we would expect a red wave and then a green wave of cells at some point, but we didn't know exactly when. So this was our work hypothesis, and everything was completely different when we actually did these transplants, because the only cell population that did the entire engraftment since day 5 actually onwards was actually the CD90 population, as you can see here in green, and the blue and red never came up in the transplanted animals. We rotated the colors, always the same result. What is good for gene therapy, because we can actually reduce the number of cells by up to 20-fold if you do the calculation, because if you just modify CD90 cells here, you're ending up with like 5 to 7% of the CD34 positive cells. And last but not least, the most surprising to us was actually, and that's something that people have done and tried with CD34 positive cells. They were trying to predict the outcome of transplantation with actually little success. It's not really well correlating with the neutrophil and platelet recovery in patients. But in these animals, when we did this and calculated the number of CD90 45RA negative cells per kilogram and plotted this against the day of neutrophil and platelet recovery onset, it gave us an almost perfect correlation. So we could actually predict the onset of hematopoietic recovery in these animals. So this is very brief. So if you want to read up on this, this is all in this 2017 paper on top. We also, because this is all based on colors, so it's not really meaningful for any therapeutic applications, I've been working with a coworker in our lab, Olivier, who is interested in treating hemoglobinopathies. And so we did a study and using CD90 for the treatment. So just briefly, as a proof of concept that it's not only something we can do with a GFP and a cherry. So we used sickle cell anemia in the lab as a model in order to mimic what has been actually successfully done last year in the patient, because this disease is highly prevalent. I think it's the second highest, most prevalent after HIV right now. And it's known actually since the 90s that there's a subset of patients that has a mutation in the hemoglobin repressor, the fetal hemoglobin repressor area, that leads to a consistent activation of fetal hemoglobin and a longer survival. So there's two different approaches out there that people are following. The one is by keeping the fetal hemoglobin expression up by basically knocking out the repressor, BCL11A. What we don't like, because there's also some cross-reaction in other tissues, we actually prefer doing inducing a mutation in the binding side of this repressor. So basically not blocking the binding of the repressor and therefore activating the fetal hemoglobin expression. And that's possible since we have CRISPR. So we did this, as I mentioned again, in the monkey model. Just here briefly outlined our transplant strategy. It's just, if you want to read up on this, we have a lot of publication on this. But the key of this experiment was we did this again on CD34 positive cells, the current gold standard, where this is our CD90 positive HSC enriched fraction, and compared this in the monkey. And we transplanted this and followed these animals longitudinally. So I'm only showing you the gene editing data, not the hemoglobin data, because of problems with time here. So blue, again, are the CD34 animals, and on the y-axis we have now the percent of gene modification in the peripheral blood. And you can actually not see any difference in between the CD90 as well as CD34 and CD90 transplanted animals, telling us that CD90 purification does not have a disadvantage. It only has the advantage of reducing the cell number by more than 10-fold, 15-fold, with an identical outcome. I just want to draw your attention to this very first 10 days here, because we did something that is more likely happening in the clinical application. We have so few CD90 cells, and we were concerned that if we don't co-transplant the CD90 cells that were unmodified, that something is delayed in engraftment. At this time, we didn't know that you could actually transplant them alone. It would be sufficient. So we basically modified CD90 cells, co-transplanted with CD90 negative cells. And so you dilute the cells, and so all the red lines drop to a very low level, and then jump up to like the high level where the CD34 animal is indicating again that the CD90 subset takes over entirely within a very short period of time, replacing everything, and that this is the important population that needs to be modified and transplanted. So, I mean, these are all experiments that we did in a monkey model. We did this all with the vision to translate this actually from a preclinical model into really clinically feasible application. So what we call from bench to bedside. So, and we are not the only ones who are doing this. I mean, there's other groups out there, there's other markers out there, namely CD133, what is a subset of the CD34 cells, or CD38 low negative cells, which is also a subset of this 133, and CD34 that's even smaller, and then our CD90. But if you quantitatively compare this, they are subsets of each other, not perfectly resembling each other. But when you run the numbers, the CD90 has the least number of cells for modification. So just quantitatively would need the least amount of modifying agents. So this is just a theoretical play. How do we actually do this practically? So CD34 enrichment is commonly done with a bead enrichment, and it's well established. I mean, we have the the Clinimax, the old one, as well as the new Prodigy that is basically doing everything fully automatic in a closed system. You can do the same for CD133. It has actually been done in some studies for solid tumors, lymphoma, neuroblastoma, or other heart diseases. The CD38 is a little bit different. There's two different groups out there. There's one bead-based strategy that would basically piggyback on the Clinimax bead enrichment, where you do a depletion, where you can get different levels of depletion. But you can also sort purify these cells, like the Italian group around Luigi Nardini and Bernard Gentner have been proposing. And then last but not least, our strategy where we propose to combine the Clinimax Prodigy as a closed system with a sort purification. And at this point, when we started this project, we were pretty much unbiased. We didn't really know which system would be ideal to use. So we actually had at this time a new Sony cell sorter available in our GMP facility that has the ability to replace the entire fluidic system. But we also had the Tyto from Miltenyi that is a cartridge-based, fully closed cell sorter with a chip on the bottom. So we designed a very simple experiment, simple but expensive and really complex. So we took a leukapheresis product of healthy donors, GCSF mobilized, enriched CD34 positive cells on the Clinimax. So basically at this point you could say, well, that's a product that would be commonly used. It split this into 3 fractions, kept 1/3 as bulk CD34, 1/3 was sorted on the Sony for CD90, and another one was sorted on the Tyto for CD90. So basically the 3 different blocks on the right side. The products, the CD34 bulk, as well as 90 from the Tyto and Sony, were then transduced with the lentivirus again with GFP, and the cells were then introduced into multiple readouts. And I'm just going to show you a little bit of this data. All of this is actually publicly available on bioRxiv. We're just right now in the revision phase of this paper. So one of the crucial questions initially is how good is the product, just by purity and yield. So we have a good purity on both systems. The Sony and the Tyto perform pretty much evenly. To BD area, it's pretty much identical. I'm using not really a purity setting. I'm actually going more for a yield because the cell, I don't want to lose anything. That's why I'm only getting up to like 90% purity. So there is a little bit of contamination, but we know that these contaminations are not harmful because 90 negative cells have been transplanted for years without problems. The yield looks not ideal. It's about 50-60%. That's because we have been really conservatively gating. So the CD90 expression is very low, so you have to be really careful how you gate. So if you gauge too low, you get a lot of CD90 negative cells, and the purity will be really, really bad. So we had to find a compromise and go for the CD90 high expressing cells, ending up with a fairly decent 50-60% yield. And the most important one, actually, as I mentioned initially, our prediction was that we actually do an enrichment of 10 to 20-fold. But if you run the numbers, we're actually more in the range of 30 to 40-fold enrichment, what is much, much better. So again, savings are then also higher. Then we introduced these cells to the virus and we transduced them. And then we were very surprised to see that the pure purification of the CD90 subsets itself without any transduction enhancers or any changes other than the sorting in our standard transduction protocol that we're using in clinical strategies increased the lentiviral transduction efficiency by 2 to 3-fold. A little bit higher on the Sony than on the Tyto, but significantly higher than the CD34 CD90 subset in bulk CD34 positive cells. What was also surprising to see is people would be now concerned, okay, we're exposing these cells directly to the virus, so you would expect more transduction events per cell, and it could increase the risk for oncogenic events. But this is actually not the case. We actually have still the same average of vector copy numbers for the cells when we look on single colonies after modification and 2 weeks of CFC culture. What is very surprising, because there seems to be a cell inherent mechanism blocking these cells from getting overly transduced from more than multiple times when they are directly exposed. And then last but not least, these transduction experiments are done worldwide very often, and the majority of people are reporting really excellent efficiencies, even higher than 60% actually. But it's really hard to translate this into an in vivo situation. In most cases, we see a very high efficiency in the very beginning, but then they taper off and go down, like we have seen in the hemoglobin study. So we actually transplanted these cells into mice to assess, first of all, their engraftment potential, multilineage differentiation output, as well as then the GFP level in these animals. I'm just showing here the CD34 cells. They engraft just fine. They're adult NSG mice and adult cells, so we expect something around 5 to 10% in average. The Sony cells perform a little bit less well. It's like slightly lower, but the Tyto cells are really well in grafting. And the bone marrow is nicely reconstituted for the Tyto sorted cells as well as the CD34. The Sony cells were not performing as well. There's different symbols. We did this in a triplicate with 3 different donors, so you can see like little squares, little spheres, as well as triangular structures. But the most important part was actually that we could basically see what we see ex vivo, also in vivo. So we saw a much higher transduction efficiency and in vivo gene modification in the animals that received CD90 sorted cells from the Sony and Tyto in comparison to CD34, not only in the peripheral blood, but also in the bone marrow, what is really, really encouraging. Last but not least, looking a little bit more in detail in the bone marrow, we want to see that the CD90 cells actually engraft and that these CD90 cells are transduced. And that was definitely the case for both for the Sony as well as the Tyto sorted cells, and much better than the CD34. As you can see here, there's multiple animals that barely have any engraftment of CD90 because it's just a random and stochastic event. We introduced these cells after the harvest of the bone marrow again into CFC assays to see if these cells are actually still potent to grow out in colonies. And the Tyto cells performed really well in the CD34 and CD90 sorted cells. On the Sony, it was a little bit on off. So just let me summarize at this point. So what is our ideal future looking like for stem cell gene therapy? We just jokingly said it could be stem cell transplant 3.0 after crude bone marrow and CD34, we could get into the 3rd generation. So ideally, we would take a patient and mobilize with either GCSF, AMD, or other agents that are out there, do a leukapheresis, purify on the CD34 on the prodigy, introduce these cells into the Tyto to keep it a closed system, engineer these cells in the prodigy again, because there's a chamber that allows us to do a gene modification in a closed system again, and reintroduce these cells then into the patient. So what I've shown you so far is we have a new target that allows you to actually basically get a high efficiency, save reagents, and improve the modification, at least for lentiviral vectors. For the nucleases, we also have a more advanced strategy over the previous one, where we use electroporation, where we see a much better modification in CD90 now. We have an enhanced predictability and a much more reproducible outcome also in the animals. And the other thing is we see no clonal dominance in our monkey model as well as in the mice, but it's actually telling us it's actually safe to modify these CD90 cells. There's something I haven't shown you. All the monkey studies were done in an autologous setting. We also did an allogeneic setting with Leslie Keane together. This data was actually accepted yesterday for publication, so it should be out in a couple of weeks. We can do this with different mobilization regimens. GCSF, AMD works, newer drugs also work pretty well. And this whole system is disease independent. So I'm not focused on a single disease, but a disease like HIV. I can apply this to any disease that can be treated with stem cell gene therapy. Last but not least, combining the prodigy and the Tyto would allow us to make this a portable and closed system, but also makes a GMP facility independent, and you basically make it accessible to countries that usually don't have the infrastructure. because our overall goal actually of this work is to make gene therapy affordable and, most importantly, accessible to everyone around the world, what is currently just not the case.